Hello viewers, I welcome you all in this last session on carbohydrates. In our today's session, we will first briefly review what we had done in our previous sessions, and today we will talk about disaccharides, especially structure of maltose, lactose and sucrose. Then we shall talk about polysaccharides, where we will discuss about the structure of starch, cellulose and glycogen. Finally, we will briefly talk about the sweetness of sugars, and then we will summarize our today's work as well as the total work on carbohydrates. Till now, we have discussed in carbohydrates that there are certain compounds which are known as monosaccharides, which contain aldehydic ketonic functional group and hydroxyl groups. Amongst these, we have discussed in detail the structure and configuration of glucose, which is a very common monosaccharide. Then we discussed that these monosaccharides exist as a cyclic structure. We discussed that how the structure of these compounds is determined, initially for their glycosides, and finally with the help of certain instrumentation techniques the structure of these monosaccharides are determined. Now next we talk about disaccharides. Disaccharides are carbohydrates that are made up of two monosaccharide units. That is, hydrolysis of a molecule of disaccharide, it will yield two molecules of monosaccharide. The two monosaccharide units are joined through an ethereal or oxide linkage called glycosidic linkage. We have earlier discussed in detail in one of our previous session about the glycosidic linkage. We discussed that these monosaccharides, when they exist as cyclic structure, that is like an hemiacetal, and when they react with alcohol in presence of acids, one molecule of alcohol is consumed and they give structures which are acetal like structures, and they are called glycosides. Then these glycosides have obviously two parts. One part is from the sugar part and the other part is called as aglycone, which can be an alcohol or phenol or maybe some other sugar moiety also. We have discussed this thing earlier, that if this is the hydroxyl group at the anomeric carbon, which is formed after the cyclization of the ring, reacts with alcohol, we get these compounds which are called methyl glycosides, and this linkage by which this group is attached to the sugar ring is called glycosidic linkage. Then, as I said, that it is not always necessary that other part is only alcohol, it can be another monosaccharide moiety, because it has got many of the hydroxyl groups. One of the hydroxyl from monosaccharide may combine with the hydroxyl group present the anomeric carbon of another saccharide, and they may give a disaccharide. Like this is a molecule of alpha D glucopyranose, this is another molecule of alpha D glucopyranose. This is the hydroxyl group at position 4. So this hydroxyl, if it acts like an alcohol, and this is the hydroxyl group at the anomeric carbon, they may react, eliminate a water molecule, and they are joined now by a glycosidic bond or glycosidic linkage. Such a unit is a disaccharide, because now it is made up of two monosaccharides. But amongst these two monosaccharide moieties, we will call this as having the glycosidic linkage, or this is being converted into the glycoside, and this is the aglycone part, because this is acting like an alcohol. Then linkage may be formed, as I said, that hydroxyl group at the anomeric carbon of one monosaccharide or both the monosaccharides also. The another possibility is that suppose this is one sugar moiety, this is another sugar moiety, that is monosaccharides, and they may form this ethereal linkage or the acetal bond using the hydroxyl group of anomeric carbon in both the cases. So now both are basically having the glycosidic bond. If I call this monosaccharide as a glycoside, this is aglycone, or this as a glycoside, this is aglycone. So we will see that such disaccharides can be named in two different ways. Again, this is an example showing with the conformation of any monosaccharide. Say this is one sugar, Now, this is the anomeric carbon having the hydroxyl group. When it reacts with any alcohol in presence of acid, it forms this acetal type of structure, and we call this bond as a glycosidic bond. Now, this is the glycoside of this monosaccharide with an alcohol. Now, suppose it reacts with another monosaccharide unit where this is carbon number 4 having this hydroxyl, this combines with this. Now, we get this unit. This bond is called a glycosidic bond, and this unit is called a disaccharide. Let us take the example of disaccharides. One of the example of disaccharides is maltose. Maltose is hydrolyzed with dilute acids or the enzyme maltase to give 2 molecules of D plus glucose. Means a molecule of maltose consists of 2 molecules of D plus glucose. It is a reducing sugar. If you remember, we had discussed earlier that those monosaccharides or disaccharides which reduce Fehling solution or Tollens solution are called reducing sugars, and it is due to the presence of the aldehyde group or the free ketonic group in them. This is the structure for maltose. It consists of 2 glucose moieties. This is one glucose molecule which is using its hydroxyl at the anomeric carbon for the glycosidic bond formation. This is another glucose molecule which is using the hydroxyl at carbon number 4 for this bond formation with it. So, we call this as the alpha linkage. This glycosidic linkage is called alpha linkage because the monosaccharide unit that has formed the glycosidic bond is using the hydroxyl group which is of the alpha ionomer, or we can say that hydroxyl group is used from that side which is representing the alpha ionomer of it. So, we also call it as the alpha linkage, and this is another monosaccharide molecule which has used its hydroxyl group at carbon number 4, but its this side is like hemistal, and this ring can be opened here and aldehyde group can become free. That is why it behaves as a reducing sugar. But if we take 2 rings, this is the non-reducing ring, whereas this is the reducing part of this disaccharide. This is the beta ionomer as shown, and similarly here this is the alpha ionomer which is shown, because now here the ring can open and cyclize and 2 ionomers will exist in equilibrium, and as we have discussed earlier, the hydroxyl in the Haworth projection goes downward in the D series of sugars. It is the alpha ionomer and this is the beta ionomer. Now, how these disaccharides are named? We are naming this sugar moiety. This is glucose. It is D-glucose. 6-membered ring structure, so it becomes D-glucopyranose, and it is the beta ionomer which is shown here. So, it is beta D-glucopyranose, which has got another glucose moiety attached at the carbon number 4. So, we call it as 4O. O indicates that linkage is through oxygen, and what is linked here? Another glucopyranose molecule through the alpha linkage. So, we call it as 4O alpha D glucopyranosyl. This is like as we are using the word glycoside, and then it becomes the glucopyranosyl, and this is the beta ionomer beta D glucopyranose. This is the full name, and this is the alpha ionomer. So, only difference between the two is 4O alpha D glucopyranosyl alpha D glucopyranose. So, this way disaccharides are also named, but they are more popularly known by their simple common names, as the simple name for this compound is maltose. We can show the 2 ionomers in their conformations also. This is again the beta ionomer with the hydroxyl on the upper side, it follows the equilibrium position, and this is in the axial position. This is the alpha ionomer, and other groups are as the positions we have discussed earlier for D plus glucose. Here it is the hydroxyl at the 4th carbon which is going downward, and this is the alpha linkage from this glucose moiety which hydroxyl was also downward. So, the linkage is shown like this. This is the glycosidic linkage. Next disaccharide we take is lactose. Lactose is present in milk and it is also a reducing sugar. It is hydrolyzed by dilute acids or by the enzyme lactase to give D plus galactose and D plus glucose. Means the two monosaccharides which constitute lactose are D plus galactose and D plus glucose. Now how they are linked? They are linked like this. It is the fourth carbon of this glucose moiety. Its hydroxyl is linked with the anomeric carbon of the galactose moiety. So it is the galactose moiety and having a beta linkage. This hydroxyl belongs to D series and hydroxyl is going upward here. Now the bond has been shown like this, that we are taking a bent line from the lower side and the bent line from the other side. It is just for the convenience to show. Actually these molecules will occupy such a position so that their bonds are there between this carbon through this oxygen. And when this hydroxyl group of this glycosidic bond is linked from this side, then we will call it as a beta linkage. They will be actually represented like this. This is the galactose moiety. Its hydroxyl at this position is going upward. This is another glucose moiety whose hydroxyl at carbon number 4 is coming downward, and they have formed this ether linkage or glycosidic linkage here. This is the non-reducing half because it is like an acetal here, whereas here this can open up the ring, can be converted into aldehyde group, and this is the reducing half. But overall this is a reducing sugar. We can show its conformation like this. This is the fourth carbon of the glucose having this galactose moiety. So it is named as 4O. And now what is the galactose part which is attached? It is beta D galactopyranosyl, which is attached through oxygen to the 4 position of what? D glucopyranose. Now this is the alpha anomer which is shown. So we may introduce here the alpha also, that it is alpha D glucopyranose. If the hydroxyl is going on this side, the another anomer, it will be beta being introduced here. Because this ring can open up, lactose will also exist as alpha and beta, both the anomers. Then another disaccharide is sucrose. Sucrose is the commonly used table sugar that we generally used at our homes. Sucrose is also hydrolyzed by acids or by the enzyme invertase. Next disaccharide we take up is sucrose. Sucrose is commonly known as sugar and it is the most commonly used sugar, also called as table sugar at our homes. Sucrose is hydrolyzed by acids or by the enzyme invertase, and it on hydrolysis gives a mixture of glucose and fructose, and this mixture is also known as invert sugar. One very common example of invert sugar is honey. Now why do we call it invert sugar? Actually sucrose itself is a dextrorotatory molecule. When it is hydrolyzed, it gives equimolar mixture of glucose and fructose. Out of these, glucose is dextrorotatory, whereas fructose is levorotatory. And the value for the optical rotation for glucose is plus 52.7 and that for fructose is minus 119. Now what happens when we get the equimolar mixture? That net resultant is some negative value. So it is said that we start with the sucrose which is dextrorotatory and we are getting another product which is sweet, is a mixture of glucose and fructose, but it is levorotatory. So it was given initially the name invert sugar when nothing about the structure of glucose, fructose, etc. it was known at that time. But there is no inversion of configuration or anything else is going on in this. Then this is a non-reducing sugar. Means now here the two molecules, that is glucose and fructose, they are linked in such a way so that the anomeric carbon is not free. It means obviously they are linked through the anomeric carbon. A molecule of sucrose gives a mixture of glucose and fructose and they are linked like this. This is one molecule of glucose which is linked by the alpha linkage, means its alpha anomer is used. whereas this is the molecule of fructose. Now see, in fructose molecule it is not written in the usual way in order to show the linkage. This is the carbon number 2, which is the anomeric carbon here. So in this case what has been done, suppose in Haworth projection this hand is representing the fructose unit, here is the oxygen, on this side should have been the carbon number 2. So for showing the linkage of the hydroxyl at carbon number 2, we have written it in this inverted way. Means it is another horizontal plane, the carbon number 2 that was on this side, we have inverted in such a way so that this we are seeing here in this structure carbon number 2 on this side. So if hydroxyl here was going down for the alpha ionomer, then it will be going upward for the alpha ionomer here, and since this fructose is linked from the lower side, it means it is the beta ionomer. So be careful about remembering this, the number, it is carbon number 2, 3, 4 prime from this side, and this is the beta ionomer of fructose linked with the alpha ionomer of glucose. So that is why we say on this side the glycosidic linkage we call it as a alpha linkage, and on this side this glycosidic linkage we call it as a beta linkage. And the name for this disaccharide now, it becomes alpha D glucopyranosyl and beta D fructopyranoside, or we can name from this side that it is beta D fructopyranosyl alpha D glucopyranoside. So we can name from either side, we can take this as an aglycone part or this as the aglycone part. Better way we can show the linkage when we write these sugar moieties above and below and their conformations also. In the alpha ionomer of this glucose, this hydroxyl is downward, and in the beta ionomer of the fructose, this hydroxyl is upward. So they are written this way so that we can see clearly the linkage like this, that fructose is using the beta ionomer and glucose is using the alpha ionomer, and anomeric carbon of both the monosaccharides are blocked, the ring cannot be opened and they will behave like a non-reducing sugar. Now let us move on to polysaccharides. Carbohydrates which yield a large number of monosaccharides on hydrolysis, we call them as polysaccharides, and in our routine life we deal with more of the polysaccharides. Most of the common examples are starch, cellulose, glycogen, different gums and pectins, etc. Most of the carbohydrates encountered in nature are polysaccharides, and two main functions of the polysaccharides are that they are used for the food storage and to provide structure. The food storage may be in plants or may be in animals, and basically they provide structure to plants. Let us talk about the most common carbohydrate is starch, and this is the most important source of carbohydrates in human diet. It mainly occurs in sweets, roots and tubers. Seeds like maize, wheat, rice, roots like potato, etc., they are the rich source for starch. Starch is basically a polymer of alpha D plus glucose. It consists of two types of components. One is called amylose and other is amylopectin. Amylose. This part of the starch is water soluble and it constitutes about 20% of it. It is a straight chain polymer in which about 200 to 1000 alpha D plus units are linked. Glucose units are linked by alpha glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. We had discussed earlier maltose as a disaccharide. Actually it is, you can say, maltose extended on each side with the same type of linkage with another alpha D plus glucose ultimately gives us amylose, or amylose when it is hydrolyzed up to the 2 monosaccharide unit fraction, that will be maltose. This is the linkage. Suppose this is one glucose moiety, its hydroxyl and the anomeric carbon is downward, so it is the alpha ionomer which is being used. It is linked to the hydroxyl at carbon number 4 of another glucose moiety, and its hydroxyl at carbon number 4 is linked to the anomeric carbon of the another glucose moiety, and here also it is alpha linkage. So, this way the chain is built up, and the chain is built up like this, that the anomeric carbon of one glucose moiety, that is we call it as C1, it's linked to hydroxyl group at C4 of another glucose moiety, and linkage is the alpha glycosidic linkage. We can show in the form of conformations like this, that this is the chain of glucose molecules, and it is the carbon number 4 of one glucose moiety which is linked to carbon number 1 of another glucose moiety, and this hydroxyl is downward. So, it is the alpha glycosidic linkage. So, all the glucose moieties are linked by alpha linkage and 1:4, that carbon number 1 is of one glucose is linked to carbon number 4 of another glucose molecule. Amylopectin, it is insoluble in water and it constitutes about 80% of starch. So, 20% is amylose which is water soluble, 80% is amylopectin which is water insoluble. It is a branch chain polymer in which about 300 to 6000 glucose units are linked, and the average chain length is about 25 glucose units. The glucose units are linked in the same way as it was in case of amylose. They are having alpha glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. But how the branching occurs? The branching occurs by the linkage between C1 of one glucose unit and C6 of another glucose unit present in the chain. Means the chain of glucose unit which is being formed by the linkage between C1 and C4, they will have the hydroxyl group at carbon number 6 which is free, and for some of the glucose units this hydroxyl group at 6th carbon joins with the carbon number 1 of another glucose moiety, and then a new branch chain starts there. Let us look at this structure. This is the chain of glucose moieties with the alpha linkage and carbon number 1 of one glucose linked to the carbon number 4 of another glucose molecule. This chain is like amylose, but here the branching has occurred. This hydroxyl group at the carbon number 6, this CH2OH, has also joined with the C1 of another glucose moiety, and now it is forming another chain with 1,4,1,4 linkage and so on. So, a branching has occurred here, and due to this branching this becomes more water repellent or they become water insoluble. We can show in terms of conformations also that this is the chain of glucose molecules where C4 of one glucose is linked to the C1 of another glucose, C4 is linked to C1 and so on, and this hydroxyl at carbon number 6 is linked to C1 of another glucose molecule which can form another chain. So, this way branching occurs in amylopectin. Next polysaccharide is cellulose. Cellulose imparts shape and structure to plants. It is an important constituent of the cell wall of plant cell. It is a linear polymer of 300 to 2500 beta D plus glucose units. Now, you see again the basic unit is glucose, but here it is the beta D plus glucose. These glucose units are joined by beta glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. We can see the structure like this. Say this is one glucose moiety. Since it is in the beta anomer which is being used, the hydroxyl is upward, and it is linked to another glucose moiety, and it is the hydroxyl at carbon number 4 which is forming bond with the hydroxyl at carbon number 1. Similarly, its carbon number 1 is forming the bond with the hydroxyl at carbon number 4 of another glucose moiety. So, glucose molecules are linked by beta linkage here, and this is the main difference between starch and cellulose. Both consist of D plus glucose molecule. The only difference is in starch there is a alpha linkage, that is it is the alpha anomer of glucose which is involved throughout, and in cellulose it is a beta glycosidic linkage, or we say it is a beta anomer of glucose which is being used up. On hydrolysis, both of these, whether it is cellulose or starch, they provide us D plus glucose, which is the main compound which gives us the energy for our work. means the sugar that we eat or the starch that we eat in our body, it is ultimately digested and hydrolyzed to glucose moiety. But our body has the enzymes to digest or to break down the bonds in the starch only, that is the alpha bonds are being cleaved. We do not have any enzyme in our body which can break the beta linkage, but many animals do have. So, that is why animals eat sometimes the leaf and other parts of the plant and they get energy from them, but we can get energy only from the starch molecules. Now, this is another way of representing cellulose molecule, where it is showing the carbon number 4 hydroxyl going downward for one glucose molecule, and this is the carbon number 1 of another glucose molecule where linkage is shown from the upper side, because it is the beta ionomer or beta linkage and hydroxyl is on the upper side when the representation in Haworth projection, and so on. This way cellulose is represented in the Haworth projection. This is another conformational representation for cellulose, in which what has been done, you concentrate on the middle glucose moiety, it is not written in the normal way. Again it is inverted, and it has been inverted in such a way, I will try to explain with the help of my hands. Suppose this is the planar glucose molecule and we said that hydroxyl is on this corner always. Now, in order to show the linkage, what has been done? It has been inverted in this way. So, now this oxygen which was here has come here, but carbon number 1 retains its position, but the hydroxyl group, if it was going upward at carbon number 1, is now downward, or if it was downward it will be going upward. Since cellulose uses the beta ionomer, obviously the hydroxyl would be upward, and when it is shown like this, now it is downward, because this is the equatorial position. Upward position will be the axial position here, where hydrogen will be there, which have not been shown just for the simplification. And this is linked to hydroxyl at carbon number 4, where hydroxyl at carbon number 4 is gone downward. So, we can show a quick linkage like this. Now, this is the carbon number 4. Here the hydroxyl would have been going downward, but since we have inverted this glucose molecule written in the inverted way, the hydroxyl is now upward, and now this glucose molecule is again in the normal way, its hydroxyl is going upward. So, we can show a linkage very easily like this. So, these are different ways in which cellulose can be written. Structure remains the same, it does not change, it is just the different ways in which we write. So, this is about the two main polysaccharides, starch and cellulose. Then a third polysaccharide is glycogen. In animals, the carbohydrates are stored as glycogen. It occurs mainly in the liver and muscles, and it is a branch chain polymer of 6000 to 30,000 alpha D plus glucose units. Means again the monomer unit is same as it was in starch, it is alpha D plus, but it is a very large molecule in comparison to even amylopectin, having about 6000 to 30,000 glucose units linked together. Its structure is similar to amylopectin, but it is more highly branched and with an average branch length of 12 to 15 glucose units, whereas it was about 25 in case of amylopectin. Glycogen is also known as animal starch, and the process of formation of glycogen in the body is called glycogenesis. It is formed, say, when we eat and our food is converted into glucose in our body, and that much glucose is not required by our body at that time. So, this process of glycogenesis occurs and glycogen is formed in our body. But when sometimes we are not getting the food and body needs glucose to get the proper energy, at that time this is broken down with the help of certain enzymes, and then the process is called glycogenolysis. So, the process of glycogenesis and glycogenolysis are continuously going on in our body. Sometimes glycogen is being produced, sometimes glycogen is being consumed. This is the representation for these three forms of carbohydrates, polysaccharides. This is amylose. This one small circle is representing here one glucose unit. they are linked in a straight chain, there is no branching, and they are coiled up or they may be present in different forms in their different sources. This is amylose. In case of amylopectin, there is a branching, and in glycogen the branching is more severe. After every short break there is a branching. So, it is more highly branched than amylopectin. That is why amylopectin and glycogen has lesser surface area exposed, can form very little hydrogen bonds. So, they are practically insoluble, large size and lesser exposed area. Whereas, in case of amylose, in the chain the hydroxyl group on each glucose moiety they are exposed and they can form hydrogen bonds with water, and that is why it is water soluble. Then we have been talking about the sugars, that monosaccharides or many disaccharides are also called as sugars. In the very beginning, we classified some of the carbohydrates which are sweet in taste as sugars and others are non-sugars. Let us compare their sweetness. The sucrose, which is the most commonly used sugar at our home, if it is given a value of 100, then in comparison to it, glucose is 74% sweeter, means glucose is less sweet than sucrose. Galactose is much less sweeter, it is 33%, but fructose is more sweeter than sucrose, it is 173. If the value for sucrose is 100, means it is 1.73 times sweeter. Maltose is again 33, lactose is 16, whereas the starch and fiber means cellulose etc., they are not sweet, they do not have any taste, but because they do not dissolve in water, they do not cause any taste. So, this is about the sweetness of carbohydrates, and with this we complete our studies on carbohydrates. Let us briefly revise what we have done. We started with the classification of carbohydrates as monosaccharides, disaccharides and polysaccharides. Then we discussed about the structure of monosaccharides, taking the example of glucose and then fructose. We discussed some of their chemical reactions on which basis their structure was elucidated. Then we found that they do not exist mainly in the open chain structure, rather they exist as cyclic structure, and when the cyclic structures are formed, then the aldehydic carbon or the ketonic carbon can give two new anomers. They are the stereoisomers of each other and they differ in their configuration at the newly formed asymmetric carbon atom. Then we discussed about the string size of the carbohydrates, particularly monosaccharides, in the form of their glycosides. Then we discussed the structure of some disaccharides today, and then we discussed the structure of maltose, lactose and sucrose. We talked about polysaccharides, we discussed the structure of starch, cellulose and glycogen, and finally we compared the sweetness of some sugars. Thank you.